thank you very much, Angela, for joining us today. Um, uh, Angela Cenci Nilsson is in Lund University, which for those who don't know, that's Lund University behind Angela's head, and it's in southern Sweden, and has an amazing reputation in the world of Parkinson's disease, having done an amazing amount of work. Many people would know it for its cell therapies, but Angela has pioneered the work in dyskinesias, the involuntary movements you see in Parkinson's, often in response to medication. So you're very welcome, Angela. Thank you very much for coming. You always make me look very shabby. That's not particularly difficult, but you're always very smart. And I thought it would be useful as we just to have a little bit about your background. You know, where were you born? You know, what were you doing as you were growing up, and sort of a little bit about how you ended up in Parkinson's disease at the end.
So first of all, thank you so much for having me. I'm very, very happy to connect with the WPC community. First of all, I am Italian, and I grew up in a small town in northeast Italy. The town is named Noventa Vicentina, for those who may travel through those areas.
Northeast, you say?
Northeast is the Veneto region, region of Italy. And I grew up in a non-academic family. Then the deal was that after high school I would get the job to earn some money, but I didn't want that, and I decided to study medicine, and I did that at University of Padova and Verona.
That's quite near. That's in northeast Italy as well.
It's northeast. It's in the Veneto region. So then I graduated, and then I entered a residency program to train as a neurologist. And I started very early during that time to combine clinical activity with some work in the lab. And then I thought that, at that time, the Italian newspapers were describing these pioneering studies where immature dopamine neurons are transplanted to the brains of adult rats, and these neurons survive and they release dopamine. Well, that wasn't yet known, but so I said, okay, I would like to have some research training in that nice place where they do these experiments, and that place was Anders Björklund's laboratory at the University of Lund. So initially, I got a scholarship for a training period of 2 years, still having connections with Verona, going back and forth some periods. And then I decided to stay and say, now we have to do research the hard way. Now I go for a PhD in Sweden. And then after that came, yes, please interrupt me.
So what did your parents think of this? Because they wanted you to get on and earn some money, and you were pushing off to a medical school.
So first of all, I don't want to, my parents were wonderful people, and the only concern was, of course, the economical concern. Of course, they were kind of puzzled when they heard that I wanted to move to Sweden. Nobody had ever done that. They didn't know how this would end up, how this would turn out. But at some point, I remember this sentence from my dad. He said, now at the point where you are no longer able to give you any advice. So you have to decide what is best for you. And that was a fantastic thing to say. It gave me the courage to pursue my goals.
And when you went to medical school, obviously you were training as a doctor, but very early on you were taken by the research bug, really. It wasn't that you just wanted to practice and see lots of people with Parkinson's and treat them. You actually really wanted to get stuck into research from a very early stage in your medical career.
Well, not very early in the medical education, because again, out of economical reason, I had to finish the studies as soon as I could. But I thought that doing experiments is wonderful, because it means you are formulating a question, and then with your hands you do a job, you do some work that will give you the answer to that question. So it's so fascinating that you are creating knowledge with your ideas and with the work of your hands. The experimental work in the lab attracted me.
And you decided to go to Sweden because you were rather taken by the work of trying to repair the brain by transplanting dopamine cells into the brain.
Yes.
It wasn't that there wasn't any research you could do in Italy. It was very much the positive pulling you to.
Of course. Okay, I had started to do it in Italy, but of course, it's very important to be in the right environment, to be surrounded by people who have experience in the topic, in the methods, and give intellectual and technical inputs. So by being, if I had remained in Italy, things would have moved very slowly, moved on very, very slowly. So it's important to be in the right place.
And also with the work that was being done in Sweden, which was transplanting fetal dopamine cells and then subsequently stem cells, that's a little more controversial in Italy, I would think, with the Catholic Church and the sort of ethics around that, which are not insubstantial. But I would imagine it's harder to do that type of work in Italy compared to Sweden.
I don't know, because at some point I decided not to do this work anymore. So I don't know how the situation is today. But of course, there are many researchers doing stem cell research, stem cell work in Italy.
Yeah. So you moved to Sweden. You started working in the lab of Anders Bjorklund, that many people know here as a sort of father of brain repair in POG disease. And your research initially was on dopamine transplants.
Exactly. And noradrenaline cell transplants, dopamine and noradrenaline cell transplants.
And what was the work sort of trying to address then? Because obviously we know that if you put dopamine cells in, they can repair the brain, but noradrenaline, that's a little unusual.
So at that time, we are talking about the early 90s, how the cells integrate in the brain wasn't quite understood. So we had to show that they could not only survive histologically, so when you look at them in a post-mortem, but also that they were able to release these neurotransmitters that they can release, and that they could do that in an appropriate way. For example, when an animal is more active, there is a larger demand for release of noradrenaline and dopamine in the brain. So you want to see that the transplants can meet these demands. So there is a modulation of the release of neurotransmitters from the dopamine and the noradrenaline transplants, which we found was the case. So
you transplanted them in, and then you basically could collect or monitor what they were releasing.
Yes.
Moved around, they released this. You didn't have to stimulate the cells to release it.
No. In that time, we were using, I also did some electrical stimulation of the brain, but for completely different reasons. We were using a technique called intracerebral microdialysis, which is a technique where you can measure the levels of any chemical almost in the brain, the extracellular fluid of the brain in living, moving animals. And we were seeing the modulation with this technique, the modulation of dopamine noradrenaline release during behavior.
So basically you have an animal, you transplant the cells and you pop a little cannula in, the little animal runs around, and you say these transplants are now collecting the fluid, and I can say, well, I can see this, they're releasing it. So that was an evidence that these cells were not just pumping out transmitters, they were actually doing it in a modulated way.
Yes, they were, of course, not perfectly, but they were certainly modulated, certainly.
Yeah. And they wouldn't release it unless they were moving. They wouldn't release it when moving unless they had a connection to tell them to release it.
Yes, so it is something that people do not study well enough, I think, that the modulation of dopamine and noradrenaline release in any given brain region is regulated also at the axon terminal level. So in other words, you don't need to put the dopamine neurons in the substantia nigra in order to ensure that there is modulation of dopamine release. You can place them in the striatum, and the cells in the striatum, the cholinergic neurons, the glutamatergic terminals from the cortex will regulate to some extent the release of dopamine from the transplants. And these things should be studied better.
So basically, in POG disease, you obviously lose dopamine cells in the nigra and they project up. What you're saying is I can put them where dopamine works. They will make connections locally. They will receive connections locally. And you can, and they will release dopamine in a sort of physiological way, even though
to large extent, yes. Yeah.
So why did you go into noradrenaline? Because it's not the sort of thing you associate with Parkinson's.
Adrenaline is very important for Parkinson. Is actually very important because noradrenaline neurons degenerate before the dopamine neurons. In the noradrenaline neurons are located in a brainstem region called the locus ceruleus, and that is affected very early during the evolution of Parkinson's disease. And noradrenaline is important for many functions in the brain. It's important for motor function. It is important for cognitive function. It is important for sleep. It is important for the control of blood flow. So the loss of noradrenaline is a problem, of course.
Yeah.
And the question is more difficult to address with transplants because noradrenaline, the cells that produce noradrenaline in the brainstem, they have very, very diffuse projections to basically everywhere in the brain.
Yeah.
So if you want to put noradrenaline cell transplants, where would you place them? So that's a great question. So we were transplanting in my studies, we were transplanting them in the hippocampus and testing things related to cognitive function or more basic questions.
So you're putting these noradrenergic cells in different sites and saying, so presumably you lesion the noradrenaline network with a toxin.
Yes,
and then
of
you
course.
put in these cells and you say, what recovery am I getting and what types of recovery am I getting in different sites?
Yes, I didn't do that in such an extensive way. I mean, but that was the idea.
Okay. Yes. So you're working away there, looking at noradrenaline and dopamine cells, modulation, how they release their neurotransmitters, measuring it. And then you decided to move into a slightly different field. So what was the sort of thinking behind, I've done this, I want to move somewhere else, and why did you want to move into, I don't know if it was dyskinesias at that point, or why did you just want to move?
Well, it is because of several different reasons. I can give you one. We made some serendipitous observations that we created this Parkinson model. So in rats, meaning we were giving rats a toxin that kills dopamine neurons to recreate or reproduce the very severe loss of dopamine that is typical of the clinical stages of Parkinson's disease. And then we were treating these animals with dopaminergic drugs. Yeah. And at some point there were these behaviors occurring and that nobody apparently had reported in the literature. So these animals were showing quite abnormal behaviors. They were, the idea was the drugs will restore the movements. Mm-hmm. And what we were seeing was, the animals were moving, but the movements were very abnormal. And they had, and I said,
so it was because normally the way the
what is
works.
This? Yes, and the question was, has anybody described this? And nobody had described it. So we connected the dots and thought this must be the equivalent of what is called levodopa-induced dyskinesia in Parkinson's disease, and it was at that time a totally uncharted territory.
Yeah. So I mean, typically the model works, if I've understood correctly, you make the lesion.
Yes.
And so you have dopamine loss on one side, not on the other. Then you give drugs like amphetamine and apomorphine, drugs which stimulate the dopamine systems in different ways. And the animals spin round, and you know that you've got a lesion because they spin round. And then if you put the cells in, they stop spinning. But what you were saying is when you gave these drugs, the animals actually not just spun around, they started doing all these abnormal involuntary movements.
Exactly. They do spin around, but if you look at what they're doing, in addition to that, you have to look at the animals. So this is what people have not done, apparently. So they have these, very rapid movements of the limb all over the place that shouldn't be there. They have these dystonic torsions of the neck, of the front.
Turning their head to one side.
Yes, so they have, and they are sometimes stuck for a couple of seconds in this position. And so it was a new observation, and it was visually very striking. So when you see this, the visual impact of seeing what these drugs can do to animals was really, it really went under the skin. So I thought, okay, so now we have to find out why this is happening, and that it's.
So it was your start. Most people just put them in the tub, they run around, they collect the data, but you actually the initial observation was based just on you staring at these animals, thinking that's not right. So it was a very simple observation, which you then said, well, that's not normal. That reminds me of some of the dyskinesias that I've seen in the clinic in patients who take long-term dopamine drugs, and thought, well, are these two related, I guess, because they're a bit different, because we don't tend to give patients amphetamine in the clinic, or if we do, we probably need to be investigated. But you could see a similarity. So, so you then decided to take that on as your sort of main research area. So how did you develop that model? Because it's sort of, these animals spin very quickly. You get all these movements. You sort of something's going on, but I can't really quantify it, and I don't really know how I'm going to study this. So how did you take that question on?
Yes. So indeed, the question how do we get a quantitative assessment of these strange movements was a main question, was a very important question. I was very lucky to work with a neurologist that was visiting Professor Bjorken's lab at the time, called Sean Lee, and he and me, I was pregnant at the same time, but we were spending hours looking at the animals, and then came out with the idea that we could quantify the severity based on the proportion of observation time during which these movements appear. And having time, as the yardstick, we could grade the severity of the movements in different body parts. And at that time, it wasn't clear that this was a good method at all. It wasn't clear at all until we measured with a technique called in situ hybridization histochemistry. We measure the levels of particular neurotransmitter genes in the striatum. And I remember when I saw the figure, there is a neurotransmitter called prodynorphin. This is a technical detail, but anyway, it's expressed.
This is a transmitter in the striatum.
Is in the striatum, and it is expressed by a certain category of neurons there. Yeah. And there was a linear correlation with an extremely strong correlation. I've never seen anything like that before, between the levels of expression of this gene and the abnormal involuntary movement scores that we had recorded from the animals. And that was to me a sign. This is a method that works. So this is, give these beautiful correlations.
Yeah. So do put your questions in the chat for those who are watching if you have questions. But at this point, did you use video recording? Because I can imagine sitting there for 90 minutes or however long you're seeing these rats trying to say, well, that's a 1, that's a 2. Did you video it, then slow it down and score it like that? I mean, of course, we're used to so much technology. Yeah.
Fine. I spent many, many, many hours looking at rats, and certain subtle features like the movements of the mouth. It's very difficult to capture it with the video if you have an animal that is spinning around at the same time. So it's it was many hours of looking at the animals, but it's felt like we had an alliance with these rats. The rats were showing me things that were helping me in my research. So they were my partners in the research. So I don't think it was, I think it was quite interesting to interact with these animals.
Yeah, well, I think you make a very important point in this modern day when everything is done remotely and with technology. Using your own eyes can sometimes give you great insight. So just to recap, so you're testing these animals. They've got these involuntary movements, which look like dyskinesias. And then there's a direct correlation with this change in a chemical or a transmitter in the striatum called dynorphin. So the more dyskinesias are, the higher the dynorphin as you measured it in this particular way. So I suppose 2 questions come to mind with that. One is, had that been shown in people? And secondly, how do you know what's what? Because it could be they're just spinning around and that gives you the change in this dynorphin. Or is the change in the dynorphin driving the involuntary movement?
Exactly.
So the first question was, what was known about dyskinesias in the clinic at that point? And how could you then dissociate whether it's driving or just following the dyskinesias?
Yes. So I think that the first, if I recall correctly, the first paper showing upregulation of prodynorphin mRNA in the striatum postmortem in dyskinetic patients is a paper from Alan Crossman's group that came out, if I'm not mistaken, in 2003. And we had made this observation in 1998. So but the point is not that dynorphin is so important for dyskinesia. This is not what I want to say, is that we now know that there are thousands of genes that are expression of this gene changes in a way that is correlated with dyskinesia.
Yeah.
There are thousands of such genes in the striatum. But the principle was very important, that you have a biological change in neurons of the brain that is so very tightly linked with the movement disorders. And the link was very, very tight. So this shows we have a model that is very good to, first of all, now we know where to look for. So of course, the striatum is very important for dyskinesia. And also we have a model that is very good for further investigation. Yeah. So for me, that was a determinant observation for the choice of investing so much of my efforts at studying dyskinesia.
Because I guess one of the views at the time may have been that actually what happens with dyskinesias is you just get so much dopamine stimulation, you overwhelm the system, you get nothing changes, it's just too much dopamine. And then when the dyskinesias stop and people go off, you haven't got dopamine. So it's all dopamine and nothing else changes in the brain. And so that is the only problem. But actually what you're saying is there are clearly changes across, and with the more recent work, you're saying there are all sorts of changes across the striatum which are associated with dyskinesias. So
absolutely.
The question, so when someone's just asking about dynorphin, which is, basically just sort of a bit like an opiate, it's in the brain. There are lots of these opiate-like substances, perhaps best known in pain, but they're also found in the striatum. And they were asking about, did you see it in female and male rats? Because this is quite an interesting question. At that time, one used to use one sex of rat just because it was the easiest thing to do, but there was no real interest in male-female differences in disease and in but that's become more of an issue. So did you spot any sex differences at that stage, or was that not?
Actually, I must admit, I did not do systematic comparisons at that. I didn't do that at the time. I mean, we were only working with female rats. By the way, female patients are people with Parkinson's disease who are female have a much larger susceptibility to dyskinesia than male patients. So we were, in a way, making life easier to induce dyskinesia using female. But now that in more recent years, when we are using transgenic mice, okay, so mice and not rats, and we breed them in the lab, so we have both male and females. We see very similar changes in the female are more susceptible to developing dyskinesia. We don't know if that is because they have a smaller body weight. It could be just that, but the type of changes in the brain is very similar. So these genes that are upregulated are the strongly upregulated genes are similar. Those upregulate means the genes whose expression increases.
Okay. So now you've got this observation. So sort of turn of the century, we've got this model, we're observing it. We're seeing that we can get dyskinesia, we can quantify it. So we can now say they've got dyskinesias around their head, in one front limb, back limb, body. So you can now actually put a number on it and describe it. You've also done this clever thing where you've measured in the brain of these animals through a different technique, not through dialysis, collecting transmitters, that there are changes in dynorphin. So where did you go next, Angela? How did you then refine the model to take it to the next stage?
Well, at the beginning, when we started these studies, I was surprised to see how much resistance there was in the scientific community to accept that we could mimic dyskinesia in rodents. And indeed, we had to spend many hours, years perhaps of work to validate the model. But validate means we have to show that the model has similarities with the human disease that we claim it is reproducing. And that was a lot of work. We did several studies only for that reasons. And the way we handled the question was, what do we know about how dyskinesia is by drugs in people? Yeah. And can we test those drugs in the animals and see similar effects? So we had to test many drugs. And for example, we did dose dependence with levodopa. And of course, as expected, we found that the more levodopa you give, the faster the dyskinesia develop, the more severe they are, the larger percentage of animal will develop dyskinesia. So levodopa is clearly a very strong risk factor for dyskinesia. And this is now confirmed by all epidemiological studies in people with Parkinson's disease. So then what do people receive as a treatment if they have dyskinesia? We proceed from DBS, okay? So a person with dyskinesia would be offered treatment with amantadine nowadays. So we tested amantadine, and indeed it reduces the severity of these abnormal behaviors in the rodent. Then another drug that was tested in people and with positive results was clozapine, and that also reduced the dyskinesia. Then we tested all things that were proven in small clinical trials. This is what was available. All drugs that have shown positive effects in these small clinical trials in Parkinson's disease, and the majority of them were also working in the model. And then came then came another era that was the brain imaging investigations.
Okay. But just to sort of recap, so at the time, people were sort of saying, well, this is great, Angela, it's a lovely work. It's got no relevance to the clinic. We only see it in the clinic when people take L-dopa over years. You're giving it in a rat that's got a loss of dopamine in a very aggressive way. You're giving drugs we don't use in the clinic. You're describing something that's got no clinical relevance, but it's interesting in itself. But you're saying actually what we did was we validated the model by showing that everything we see in patients we could reproduce in the animal. So whatever, so basically I have a model for dyskinesias. And I guess the other argument which people would make, which I know we've discussed in the past, is people would say, well, why worry about dyskinesias? And I'll come back to this because I think it is a big worry and it is a big problem. All you need to do is, say, especially turn of the century, deep brain stimulation was coming along. Everyone was having it. That's going to solve dyskinesias. Amantadine is quite a good drug. And you could just give continuous dopamine stimulation. So I think Neil Quinn with David Marsden had shown many years ago that if you infuse dopamine, you could smooth people out. But of course, that is all very theoretical, not reality. So understanding how we can do it, and from a clinical point of view, I can emphasize, as many of the people on this call will, that it is a big problem, dyskinesias. I mean, obviously there are mild ones, which you don't need to worry about so much, but most people over a period of time, they do become troublesome. Amantadine is a great drug, but it doesn't work for everyone. It has side effects, and we don't have much else. So this was great that you've now got a model. You had to spend years persuading the very cynical neurologists and scientific world that you have a valid model. So what was the next major move? So you sort of say, okay, I've got a model. I can reproduce the involuntary movements. I can treat it with amantadine just like I do in the clinic, and it works. So what was the next sort of conceptual leap? Did you then want to sort of better understand, well, what is driving these dyskinesias? So can I find better therapies?
Yes. So there were two types of developments for the research in my group. One was to now that we have this model, let's use it to help identify drugs that can be tested in people. And so we had several projects funded by the Michael J. Fox Foundation, to which I'm very grateful, that enabled us to partner up with companies that had interesting compounds, and we could test them in our model. And one of these compounds was proven to reduce dyskinesia in patients in a clinical trial that was published in 2025. And that was first tested in my lab on a dyskinetic rat in the first place. So
can I just ask, when was that done? Because I think people sometimes don't realize how long it takes from an
So the first publication with the positive results of that drug came out in 2015 in the rat model, and the clinical trial it is a phase 2 clinical trial. You may want to explain what that means, but so it's not very large, but the results were really very promising, and the paper was published in Movement Disorders in August, I believe, in March 2025. Yes, and I think that with dyskinesia, the clinical development, the path from the initial observations in the lab to the clinical trial is much faster than for other areas of Parkinson. And yet it took 10 years.
Yeah. So this early work you were doing with the dyskinesias, I mean, I guess one of the questions that arises is around amantadine. So obviously clozapine, which was one of the drugs you said. So clozapine is a slightly unusual drug because it blocks dopamine, and it's quite hard to use because it has lots of nasty side effects. But amantadine has been around, since the 1950s and 60s. It's an anti-flu drug. It was a treatment for Pugsies and still is occasionally used for Pugsies. So, and when I was a young man, which is a very long time ago, one of the things they said that amantadine did was it modulated dopamine release directly. That's how it worked. It worked on the dopamine system, which subsequently proved not to be the case. So when you were able to demonstrate that mantine worked in your model, did that open up a new sort of mechanism, or how was the mantine thought to work then, and how has that helped you move into other transmitter systems like the glutamate system and such like?
Actually, amantadine is also inducing some strange behaviors in rats. So I never thought that was the ultimate drug. And indeed, you at the doses by which it improves dyskinesia, it is also inducing other strange behaviors in animals, like the animals are kind of hyperactive. And I've learned then late after testing amantadine that it can give side effects more of a kind of cognitive and psychiatric type if you give it at high doses to people. So amantadine has these dose limiting side effects, unwanted effects, meaning it is not the ultimate drug to control dyskinesia. If we want to treat dyskinesia with drugs, there is a need for compounds that not only can reduce the involuntary movements, but also generally give a better, performance in the activities of daily living that are devoid of neuropsychiatric side effects, do not cause other troubles. So this is the point that this.
Well, I suppose what I was driving at is you're absolutely right. I mean, amantadine can cause neuropsychiatric problems, people's ankles swell up, all sorts of side effects with it. So it can be very difficult to use. But I suppose the question I was after was, did you learn how amantadine worked in your dyskinetic model, which then said, well, if it works on this particular glutamate, which is another transmitter, synapse onto the dopamine cell, if I understand this pathway, perhaps I can target that. So how did you decide what to target trying to take your therapist forward? And I'd be very interested about the drug which you've taken all the way to a phase 2 trial.
Exactly. So for the drugs that was taken all the way to a phase 2 trial it's not just that it was working and we saw it was working. There was a clear investigation of the underlying mechanisms of action. And the way by what we saw was it was reducing these large peaks in dopamine release that occur in the brain every time you give levodopa to a dopamine denervated animal. So they were blunted. And this is because this is a drug that reduces the activity of serotonin neurons. So there was a clear rationale for bringing that drug further. Not just because it reduced dyskinesia in the rat. There was, we knew quite a bit about the pathways by which it works, which brings
so this serotonin. So serotonin is another one of these transmitters that comes out of the bottom of the brainstem. You've got the dopamine system, you've got the noradrenaline system where you began, which goes everywhere. And the serotonin system is in this thing called the raphe nucleus in the bottom of the brain. That goes everywhere. So why did you suspect serotonin? I mean, obviously it works very well, but why did you think serotonin was relevant for dyskinesia? Because so far you've told us amantadine worked, which I don't know that it works on the serotonin system, clozapine, which blocks dopamine, dynorphin levels have changed. So how did you get to serotonin?
Serotonin was actually an observation, a pioneering observation by a Japanese group at the end of the 90s, showing that if you have these animals with a very severe lesion of dopamine neurons and you add a lesion to the serotonin neurons, there is actually no increase in dopamine levels in the brain. And that led to a series of studies that we and other groups were involved, showing that actually in the situation where dopamine neurons are very strongly damaged, particularly if you have a lack of dopamine nerve fibers in the striatum, there must be other neurons that are able to take up levodopa, convert it to dopamine and release it. And that was the serotonin neurons, because and I can speak for a long time. So
basically the idea was you lose the dopamine.
Yes.
And so the L-dopa, which we're taking, cinnamate and matapar and such like, but the serotonin system takes this up. The serotonin neurons don't have a thing called a dopamine transport, so they can't take back dopamine once they've released it. So they were taking up dopamine, L-dopa, converting it into dopamine, releasing it. So you now have this unregulated dopamine release from a population of cells, which are not supposed to be doing that.
This is, these transient peaks that suddenly you have a peak at 40 minutes of dopamine release in the brain, 40 minutes after giving a dose, and then at 60 minutes it gets back. So these very, very transient and large peaks reflected the release that comes from serotonin neurons, and we and others could show that. So that was preceding the evaluation of these drugs in the animal model.
And serotonin, there are lots of drugs that work on the serotonin because it's obviously highly associated with depression and such like. So, so what's the story there? Is there anything about antidepressants and dyskinesias, or?
Yes, there have been studies addressing the effect of antidepressant in drugs in dyskinesia, and there have been mixed results. And I think it's a very complicated story when you work with antidepressant, because as far as I have understood, they are leading to a release of serotonin themselves that inhibits the receptor that we would need to target to reduce dyskinesia. So it's a very complicated kind of drugs. I wouldn't suggest to go that way to treat dyskinesia.
So basically the antidepressant serotonin 5-HT drugs work in a very different way to the drugs you're advocating for.
Yes, dyskinesia. Absolutely. Yes.
I mean, I think it's always fair to say, just as a point of information, anyone's dyskinesia gets worse if you get very stressed or anxious.
Absolutely. Yeah.
So in that sense, having something which calms you down would help, but it's not really what we're talking about here.
No.
And so here we are. You're starting to work up this model system. I mean, how did you interrogate it? So obviously you've talked about early on, you recorded transmitters. Then you said you did this in situ hybridization where you basically stained the brain and saw what was happening. But now you're getting into the serotonin system, and you said also in passing, there are lots of changes in the brain. So how has technologies and methodologies evolved to allow you to look in your model system in greater detail? And where is that taking us? Are there new things that are appearing through that?
Yes, of course. So I had started to say there were two types of directions that we were taking. One is to use the model to test drugs, basically to help the therapeutic development in this field. And another direction was to use the model to understand the neuronal circuits that cause these movement disorders, and which is a very, very interesting question. And this is more, it is a question of neurobiological / theoretical character, but eventually it will help also develop better treatments. So we got into the theme of how different cells in the striatum control the dyskinetic behaviors, and found that there is so in the striatum you have two populations of cells. One cell is, there are many populations, but the two largest ones are one population that whose function is to select the movement. So now this movement needs to be done. The other population is there to suppress all other movements that shouldn't occur at the same time. So we found that the population of movement selective cells was overactive in dyskinesia, and the population of movement suppressing cells was underactive. So you have this situation, levodopa comes in, it is converted to dopamine. The cells that are supposed to break the movement become, to put a brake on the movement, become underactive. And the cells that are there to select the movement, now they are active all over the place. So this will, of course, this results in uncontrolled excessive movements. And the technology used to demonstrate this phenomenon was also very interesting. The different methods could be used to address that they weren't available earlier when we started with this research.
Which is always a good point, because people say, well, why didn't you do that? And you say, well, in the 1990s and 2000, single cell, these optogenetics, none of these techniques existed. So so in early Parkinson's, so basically dyskinesia, as I always say in clinic, and you can correct me on this, is basically a function of disease duration and dose of medication. So if you give industrial doses of L-dopa early on, you'll make people dyskinetic. If you wait and give them L-dopa at modest doses, 10 or 15 years in, they'll get dyskinetic. But you're saying in the early stages, this system which accelerates or selects the movement rather, and the one that puts brake on, there's enough flexibility and buffering in the system that sort of works. So even though the drug's been taken, it's a bit crude, but it works. But after a number of years, this starts to fail. So the selection suddenly starts selecting things it shouldn't, the brake's leaky, and so now you're getting all these involuntary movements.
Absolutely.
So I guess the answer would be you could either increase some way the brake, or
Mm-hmm.
you could try and decrease the selection. So in these two populations, and I guess that's what you were doing with some of your clever modeling by switching.
Exactly. That is the $1 million question, because it's highly debated. Some people think that treatment should focus on reducing the activity of the movement selective neurons, and that's it. That's the way to treat dyskinesia. And others, including ourselves, are saying that's not enough. You also have to look at the population of neurons that put the brake on the movement. That needs to be also restored. I mean, they need to be active when levodopa is around. And it gets very complicated, because it's difficult to find treatments that do both things. And
this is different from your serotonin. Yes. So your serotonin that's basically a bunch of nerve cells or nerve terminals that are producing too much. You're now going downstream. We produce too much. So we now want to fiddle with the cells that are projecting out of the striatum. And can I selectively switch one off or one down?
Exactly. So let's say that if you think about the mechanism of dyskinesia, you have to imagine that there are different layers. So the first layer, say upstream or whatever, is that there is this peak of dopamine release every time you give levodopa, and the peak is too large and too transient. And but then the peak itself is not doing anything unless there were cells that respond to the peak. And the cells that respond to the peak are the striatal neurons, also other neurons, but the striatal neurons are certainly very, very highly involved. So what we are talking about now is the next layer, what happens to the cells that respond to the dopamine peak.
Yeah, and I guess what we're saying is that in theory, they shouldn't change because they're not affected by the disease process themselves. The disease doesn't affect those cells, but actually by giving this sort of abnormal delivery of dopamine over time, you're changing these cells in a way which is helpful to some extent, because people obviously move as a result of it, but they're producing these involuntary movements. So where would you go? So how are you going to deal with that? So what's your strategy for trying to use that? So we've got the serotonin blocking. That's great. We can reduce the dopamine back. What's your sort of approaches to try and differentiate, stimulate, or inhibit these two pathways? Is it to study them and find drugs which could work on it? Is it to try and modify deep brain stimulation?
The first thing we are doing is to try and verify whether the involvement of one category of cells versus the other category of cells differs between different types of dyskinesia and along the evolution of the treatment. So because dyskinesia is not a static phenomenon. So what you see, the first time you give levodopa, the first time you give dyskinesia, you see dyskinesia reflects changes that may not be the same when you have given the drugs for months or this is one observation. And,
so basically, just so I've got that correct, Angela, I didn't really know. So the mechanism underlying dyskinesias may change over time. So in one stage of this, and in another, it's another mechanism.
Yes, absolutely. This is what I think, but also the involvement of these striatal population, these cells in the striatum. We have data evidence that shows that the involvement of one category of neurons versus the other varies during the trajectory of the treatment. So then the question is, what kind of population is more important for a person with dyskinesia? Is it the population of neurons that puts a brake to the movement that should be targeted, or is it a population that is selecting the movement that is too active? And of course, in the absence of an answer to this, the best treatment would be a treatment that modulates both populations. And for example, the serotonin drugs are doing that indirectly because they reduce the levels of dopamine in the brain. And then we are trying to find other kinds of treatments that can do this.
But the treatments you're looking at are drug treatment. So you're trying to say, can I give a drug which could activate one and inhibit the other, so they have a bimodal action, rather than, some people will be very familiar with deep brain stimulation, where you put it in a site which is not the striatum. But I guess the question could be, could you put some electrodes in the striatum and try and fiddle with those?
We are very interested in studying also this stimulation type of treatments, but we wouldn't stimulate the striatum. The striatum is not good as a target for deep brain stimulation. It's too large. Yeah. The cells are too largely distributed over a conspicuous volume. What we are currently studying in my group is the possibility to stimulate particular type of neurons actually in the cortex, in the premotor and the motor cortex, to see if we can, which can be done non-invasively in the future or also today, and see if using that as a stimulation target, we can modulate the dyskinesia, because the cortex is strongly influencing the activity of striatal neurons.
Yeah. So there's been quite a lot of interest in that, transcranial magnetic stimulation, TMS.
Yes.
Seems to work quite well for dyskinesias, but it only works as long as you've got the stimulation over the brain. So that's always a bit of a problem. And I remember many years ago, I think Stefan Polfi had a paper in Neuron or somewhere where he put electrodes over the cortex in monkeys and then stimulated it to treat the features of Parkinson's in the monkey. I don't think it ever came to anything in people, but I can see that you're going upstream. So you've got the cells projecting out, you've got the dopamine, and now you're going up one step above. Or is it you're targeting the output and stopping the output?
Okay. Yes, absolutely. I think that there is, I don't know. So DBS in the subthalamic nucleus is a very well studied treatment for Parkinson's disease, and it is recommended to people with dyskinesia. But my understanding is that the way by which it reduces dyskinesia is mainly because it allows for a reduction of the levodopa dose. Yeah. There is the stimulation of the subthalamic nucleus per se could by itself induce dyskinesia. There are certainly other nuclei below the striatum that could be targeted for treating dyskinesia, and I'm sure there are several possibilities. I'm just saying what we are doing at the moment in my group. So yes.
But it would obviously be very attractive, because if you could stimulate the cortex, which is much more accessible than the subthalamic nucleus, it would be a much better treatment. So I just had a couple of questions related to that. One is, there's this thing called chemogenetics where people put a virus in and it expresses a receptor over quite large areas. So you could imagine, could I inject a gene therapy into someone's brain with dyskinesias, and then I can stimulate it with a drug at a low dose to switch off these abnormal pathways? I mean, I know you do that in your animal models, but could you imagine we could do that in people?
Yes, theoretically it could be done with all the risks associated with gene therapy, but of course it is something that can be done technically.
Yeah. Okay. And then the other question I had was about how much have you relied on human tissue to validate what you've seen in your models? So what I would say in my experience, post-mortem brain tissue 20 years ago, no one was very interested in it, whereas now everybody is very heavily involved with it, because you can actually study things in enormous detail at the single cell level in the human post-mortem brain. So how much has your work been informed by looking at the post-mortem human tissue? And I should say we're enormously grateful to patients and their family who donate brains, because without that, a lot of the validation you've said it would not be there. So how much have you relied on human post-mortem tissue to help support your hypotheses?
We have done something. We have collaborated with the Queen Square Brain Bank, thanks to Andrew Lees, who provided us tissue from very well characterized cases. And I remember once, this is just an anecdote, that we had a group of people with the records of having had dyskinesia and a group of people with the records of not having ever had dyskinesia. And one in the group that never had dyskinesia looked a little more, like the dyskinetic one. So I wrote to the neurologist who sent us the tissue, and I said, are you really sure this one never got dyskinesia? And he replied, yes, this was Andrew Lee's patients. We have all the records. I mean, he has his own patients in the brain bank. It was so
that's quite interesting. So you can actually use information yourself to feed back.
Yes, it's a tremendous resource, and it's a tremendous resource for research to have access to tissue donated by people who have had the disease. However, there has been also a technical development over the years where certain phenomena can be studied in vivo in the patient using brain imaging. And for example, positron emission tomography or magnetic resonance imaging, these techniques can be used, they pretty harmless it can be used in people. And there are many findings obtained in people using these techniques that confirm phenomena we've seen in the model of dyskinesia.
Yeah, so the PET, obviously you can look at receptors, and you can, so I mean, I don't know if they've got a PET
receptors, blood flow, the relationship between blood flow and metabolism. So all these, there is a phenomenon where there is an excessive increase in blood flow in certain spots of the striatum without a concomitant increase in glucose utilization that is quite abnormal, and that is characteristic for dyskinesia in the rat, and it's also seen in the humans with positron emission tomography.
And so, in the last few minutes, I think, we've obviously, I'm sure, have quite a few people on the call who, have Parkinson's, have dyskinesias. So looking to the future, I suppose what we've talked about is there are these techniques you can use, like deep brain stimulation or dopamine infusion therapies, which are helpful. Deep brain stimulation, because you reduce the amount of medication, which helps with the dyskinesias. We have amantadine, which can work, but it has side effects. And the problem in the clinic, I would say, is if people don't get on with it, we're really in trouble because there's not much else. There was this drug, Keppra, levetiracetam, which at some point was thought to be quite useful. But you've now got a new drug around the serotonin system. But looking to the future, if someone on this call was to say, okay, Angela, in 10 years' time, what will I be taking for my dyskinesias? Do you think your money's on the cortical stimulation? Do you think it's on another class of drugs?
First of all, to be prudent, I think that we shouldn't put all of our money on one kind of approach. We should let different approach develop at their own pace, because I think it's unlikely that we can find one single approach that fixes all the problems for the Parkinson's disease. And so do
you think, just on that people, a bit like when we're choosing drugs for certain conditions, do you think it'll become more personalized? So do you think, well, Angela's dyskinesias I'm going to treat with amantadine, and Roger's I'm going to treat with a serotonin blocker? Will it be like that?
Absolutely. It makes sense. Absolutely. Well, amantadine, I'm not sure, but certainly we should personalize the treatment as much as we can, provided that we have biomarkers that tell us what pathway is the most involved in a particular person. And several pathways, meaning several mechanisms, contribute to dyskinesia. They together concur to cause these abnormal involuntary movements, but the importance of one mechanism over the other different between one person and another one, and between one stage of the disease and another stage of the disease. So we need different options. Absolutely.
Okay. And do you think it'll be combination? Do you think, well, in the years to come, I'm going to give you a little bit of amantadine, reduce the side effects, give you 100 milligrams once a day. I'll give you your new drug, and I'll use low doses of both of them to get the benefits from both without the side effects. So do you think combination therapy might be the way forward?
Yes, I think combination therapy is a very good strategy to study further, and also because there are also for another reason, and that is certain aspects that are associated with the dyskinesia, certain non-motor aspects are also troublesome, and they could benefit from the same drugs that we develop for treating dyskinesia. For example, there could be an antidyskinetic compound that has antipsychotic effects, and that would normalize certain reactions that occur in the non-motor parts of the brain during under the effect of levodopa. So they would, or other kind of that are not that are induced by the medication. So for example, targeting certain types of dopamine receptors could fix several problems, and I think we haven't done enough in this field. There is a latitude for improvement. There are new molecules that could be developed that attack specific type of dopamine receptors only when they are, for example, associated with certain proteins or in a certain functional state. So there is a lot to do in that area, I believe.
So
and that could benefit.
This
is one possibility. Of course, everybody would love to find the treatment that slows down the disease,
Yeah.
And we are also trying to do that alongside the dyskinesia research. And if we found a treatment that slows down the degeneration of the dopamine neurons, of course, this will be very good for the dyskinesia too. It will delay the occurrence of dyskinesia, for example, or perhaps prevent it totally in many patients.
Well, I'm completely with you that, I obviously work on cell therapies of the brain, and someone says, well, what happens if we find a medication that stops the disease? I say, well, I'm out of a job, but I don't care because I've stopped everyone getting it. And dyskinesias will be irrelevant as a problem in bug disease if you can slow it down and cure it. But for the moment, it's not. And obviously, your work has been, incredibly informative. So it's sort of coming to the end. And I just want to sort of just have a little reflection on things because I always like to. You're an amazing example, Angela, of somebody who I very much respect in the world of science, because you haven't jumped around. You've picked a problem, you've stuck with it. At some point it was very unfashionable, at other points it's very fashionable. And I always say it's very important to pick a problem and stay at it when people think it's in fashion or out of fashion, but try and solve it. I think it's also, I would say, it's a very under-researched area. So when I was training, it was very little researched. Peter Jennering, the UK, used to do some work on it. I think David Nutt did some work in it in the States, but really there were very few people working on it. So I think what's been so impressive about your work is you've stuck with it, you've helped us understand it, and you've moved with the times with the technologies. So the technologies now give us insights which we couldn't possibly have had 20 years ago.
That's correct.
Staring at a rat was the best you could do, whereas now you could do single cell analysis.
Yes.
So it's been a fabulous talk, and your parents would be very proud of you, having left Italy from that tiny little
up there. I hope, yes, I'll see.
Yes. To come and see this. So I want to thank you very much for an incredibly informative. I've learned a lot, as I always do with this. There's all sorts of things I've learned. So I want to thank you very much. I want to thank our audience. There's lots of questions around sex differences, which I think is a very interesting area. It might be the feature of another one of these research spotlights. Why does it occur more frequently in men than women? Is there a protective effect with women? Why do women develop dyskinesias more easily, as you say? Why do men progress more quickly? There are lots of questions around that. But for today, we've really just talked very clearly and eloquently about dyskinesia. So I want to thank all of the questions we've had. I want to thank you for joining us today. I want to thank Supernus Pharmaceuticals, who fund this research spotlight. The next one of these will be in July, so it's a little while away. It's 4 months away, when we're going to be talking about the gut microbiome and Parkinson's. So that's a very hot topic. Obviously, in between, we have the World Parkinson's Congress, which will be in Arizona in May. You may be interested to know that actually the patient participation is so great that most of the slots have been taken, but there is a waiting list. So if you do want to go, do register, because we are dipping into the waiting list to bring people in. So I would like to thank you all again. I particularly want to thank Angela for an absolutely fabulous hour's conversation. I've learned so much more about you, and I've learned so much more about dyskinesias. And thank you all very much. And I look forward to either seeing you in Arizona or in July on another one of these research spotlights. So thank you very much.
Thank you, Roger. Thank you for having me. Thank you, WPC.